the cannibal brain plague. You are not just reading about a dead species, you are reading about a brain-destroying plague that swept through our closest relatives for hundreds of thousands of years and left its scar inside your DNA. Right now, today. If you carry ancestry from northwestern Europe or Papua New Guinea, there is a measurable chance that somewhere in the architecture of your genome sits a single mutation, a tiny edit, that exists for one reason only. Your ancestors were being killed by a disease that dissolved their brains from the inside, and the only way their bloodline survived was to evolve a shield against it. This is the story of prion disease in Neanderthals, and it starts with bones. In 1899, Croatian paleontologist Dragutin Gorjanović Kramberger excavated a rock shelter near Krapina and found the remains of at least 80 Neanderthal individuals. The bones were disarticulated, deliberately fractured, thermally altered, and covered in precise cut marks. Not the marks of predators, the marks of tools, the marks of other Neanderthals. The site sat largely unexplained for a century. Researchers argued over whether it was ritual, cannibalism for food during resource stress, or secondary mortuary processing of the already dead. Then, in 2003, geneticist Simon Mead and his team at University College London published a paper in the journal Science that changed the meaning of those bones entirely. Mead's team identified a genetic variant called G127V. It is a single mutation in the prion protein gene known as PRNP. And what it does is extraordinary. It confers near-total resistance to prion diseases, to Kuru, to Creutzfeldt-Jakob disease, to fatal familial insomnia. These are conditions in which misfolded proteins cascade through brain tissue like falling dominoes, destroying everything they touch, producing dementia, motor collapse, personality disintegration, and death. There is no treatment for any of them. No antibiotic reaches a prion. No immune response neutralizes one. They are not alive in any conventional sense. They are simply the wrong shape, and the wrong shape is enough to kill you. Here is what makes G127V so unsettling. It does not appear randomly across human populations. It maps with geographical precision to communities with documented histories of ritual cannibalism. Specifically the four people of Papua New Guinea, where Kuru killed thousands in the 20th century through mortuary feasting practices and isolated populations of the British Isles. For a protective mutation to spread to the frequency it has through natural selection, the death toll from the disease it blocks must have been catastrophic over a sustained period. You do not build a genetic shield against a mild inconvenience. You build it against something that was killing people generation after generation. The prion transmission route makes the Krapina bones suddenly speak with terrible clarity. Prion diseases pass primarily through consumption of infected neural tissue, brain and spinal cord. The skull-base fracture patterns at Krapina indicate precisely that tissue was targeted and extracted. And at Grandolina in Atapuerca, Spain, hominin bones from nearly 1 million years ago show identical butchery marks extending the cannibalism record deep into our evolutionary past. Researcher Yolanda Fernandez-Halvo documented those marks in the Journal of Human Evolution in 1999. They are not ambiguous. Something was eating the brains of hominins for hundreds of thousands of years before the first city existed. What the mainstream offers is a partial answer. Archaeologists accept the Krapina cannibalism evidence and acknowledge the G127V variant, but they argue the mutation may have spread for several reasons beyond prion disease: genetic drift, founder effects, other infectious pressures. Prion disease, in their reading, was an unfortunate side effect of cultural practice, not a civilization-level epidemic. But genetic drift does not push a specific protective allele to the frequencies observed without massive, sustained selective pressure. And the geographic distribution of G127V is not random noise, it is a map. It follows endocannibalism. It follows the practice of consuming the dead within your own community. Which means the question is not whether Neanderthals practiced cannibalism. We have 80 sets of bones at one site alone. The question is how many of them died screaming in the dark, confused, shaking, their brains unraveling, before their descendants evolved the mutation that let the rest of them survive. Some researchers go further. They ask whether prion disease was not incidental to Neanderthal life, but epidemic within it. A slow-burning, neurological plague, spreading through mortuary practice, incubating silently for up to 40 years in healthy-looking individuals before destroying them entirely. In that reading, the G127V mutation is not just a curiosity in a genetics paper, it is a memorial. The biological record of a catastrophe so deep in time that no one who experienced it could write it down. And if that is true, then every person alive today who carries that variant is, in a very specific sense, a survivor. Not of their own disease, of their ancestors. That disease lives in your genome. But the next one left its mark somewhere even harder to ignore. Carved directly into bone that walked the earth 1.7 million years ago. The million-year cancer. We have been telling ourselves a comfortable story about cancer, that it is a disease of modernity, of cigarettes and processed food and industrial carcinogens poisoning the air, that it is, in some fundamental way, our fault, the price paid for leaving nature behind. It took one small fossilized foot bone cataloged in a South African cave to break that story apart. Not bend it, break it. The bone is known as specimen SKX 5020. It is a metatarsal, a foot bone, and it was excavated from Swartkrans Cave in the Cradle of Humankind, a UNESCO World Heritage Site west of Johannesburg. In 2016, paleoanthropologist Edward Odes and his colleagues at the University of the Witwatersrand applied high-resolution micro-CT scanning to the specimen and found something that had no business being there. Malignant bone lesions consistent with osteosarcoma, an aggressive primary bone cancer that, in modern patients, typically kills within 2 to 3 years without treatment. The specimen dates to approximately 1.7 million years ago. It belongs to a hominin, a member of the genus Homo or possibly Paranthropus robustus. It is the oldest confirmed malignant cancer ever found in the human evolutionary lineage. The same study identified a second anomaly. A vertebra from Australopithecus sediba excavated from Malapa Cave and dated to approximately 1.98 million years ago, showed an osteoid osteoma, a benign bone tumor. Published in the South African Journal of Science in 2016, the paper confronted the field with something it had preferred not to confront. Cancer predates our species by nearly 2 million years. It predates cooking. It predates tools. It predates language, agriculture, fire, and every habit of civilization that we have blamed it on. The mainstream response is measured. Paleontologists point out that osteosarcoma can be triggered in young individuals by rapid bone growth and that the radiation environment of early Earth differed from today. Soft tissue cancers, carcinomas, lymphomas, leukemias do not fossilize, so their absence from the ancient record tells us nothing about their prevalence. Cancer has always existed in biology. It simply goes undetected because the evidence dissolves. The Swartkrans finding is striking, but it represents a pathological outlier, not a window onto widespread prehistoric cancer rates. But here is the problem with that reasoning. If we are only detecting bone cancers, the rarest and most skeletally visible type, and we are already finding them at 1.7 million years ago, then we are looking at the smallest conceivable fraction of the actual cancer burden our ancestors carried. The argument that soft tissue tumors do not fossilize is not a reassurance. It is an acknowledgement that the true scale of prehistoric cancer is invisible to us. We are not seeing an anomaly. We are seeing the tip of something enormous that sank without a trace. The bone microstructure of SKX 5020 shows aggressive trabecular destruction consistent with metastatic spread. The individual was in significant pain. Their mobility was compromised. They continued living, and presumably continued functioning within their social group, under conditions that would floor a modern adult without pharmaceutical intervention. A 2010 review by researchers in Nature Reviews Cancer argued that cancer rates in antiquity were genuinely lower because of shorter lifespans. But SKX 5020 is a young adult specimen. The age explanation does not hold here. What some researchers in evolutionary oncology have begun to suggest is that cancer is not a disease at all in the traditional sense. It may be something older and stranger, an ancient cellular program, a reversion to a pre-multicellular mode of existence encoded deep in the genome, present since the first complex organisms evolved. In that reading, the real evolutionary question is not why prehistoric hominins got cancer. It is why we evolved to suppress it as effectively as we do. What did that suppression cost us in other biological trade-offs? What did it select against? The pursuit of that question leads to places that mainstream oncology research rarely goes, because the funding structures of cancer medicine are built around destroying tumors, not asking why the program exists at all. A 1.7 million-year-old foot bone does not fit neatly into a research grant, so it gets filed. It gets cited once in a review paper, and the comfortable story about cigarettes and industrial chemicals quietly resumes. From a cave in South Africa, we cross the Atlantic to the driest desert on Earth, where 9,000 years ago, something was quietly destroying people's hearts long before anyone had a word for what disease even was. The silent heart killer. There is a parasite that enters your body without a symptom, travels to your heart, and then waits. For 20 years. For 30. It does not announce itself. It does not cause fever in its chronic phase or pain or any signal you could act on. It simply dismantles the muscle of your heart, cell by cell, year by year, until one morning the organ that has been quietly failing for 3 decades finally stops. It has been doing this to human beings for at least 9,000 years, and modern medicine still has no cure. The parasite is called Trypanosoma cruzi. The disease it causes is Chagas disease. And in 2004, paleopathologist Arthur Aufderheide at the University of Minnesota Duluth published a study that rewritten the entire epidemiological history of this organism. His team tested 283 pre-Columbian mummies from the Atacama region of northern Chile and coastal Peru, including specimens from the Chinchorro culture, the oldest artificially mummified human remains on Earth, predating the Egyptians by over 3,500 years. Using PCR analysis and immunohistochemistry on preserved tissue samples, they found the DNA of Trypanosoma cruzi in multiple individuals. The oldest specimens dated to approximately 7,000 years before the present. Let that settle for a moment. 7,000 years. In the Atacama Desert, the driest place on Earth. The epidemiological model that existed before Aufderheide's study was built on a specific geography. Chagas disease, according to that model, was a disease of warm and humid tropical zones spread by triatomine insects known informally as kissing bugs that nest in the cracks of mud brick rural housing. The bug defecates near the bite wound during feeding. The parasite enters through the skin. The disease spreads in poverty, in poor construction, in exactly the conditions that the modern developing world has not yet escaped. That model had a neat internal logic. It explained the current distribution of the disease across Latin America. It explained the demographics of infection. The Atacama mummies demolished it. The desert is hyper-arid. The Chinchorro lived in open coastal settlements, not mud brick housing. The triatomine vector that supposedly drives transmission was not established in that region during the period the mummies represent, at least not according to any known entomological record. The parasite was there before the conditions that were supposed to enable it, before the vector, before the housing, before everything that modern epidemiology had identified as the prerequisite for its existence. A 2000 study by Gould and colleagues confirmed Trypanosoma cruzi in additional Andean mummy series and identified the specific strain lineage known as TC2 as associated with the most severe cardiac manifestations in modern patients. These were not mild infections. The anatomical evidence from multiple Atacama mummies included megacolon and megaesophagus, the pathological enlargement of hollow organs that occurs in the chronic phase of advanced Chagas disease. These individuals had been living with this parasite for years, possibly decades. Their bodies bore the same structural damage that modern cardiologists spend careers trying to slow. Today, the World Health Organization classifies Chagas as a neglected tropical disease affecting approximately 6 to 7 million people globally. There is no vaccine. The only drugs available, benznidazole and nifurtimox, work only if administered in the acute phase, which is typically silent and unnoticed. By the time most patients are diagnosed, the chronic phase has been underway for years. The heart damage is irreversible. Medicine manages the decline. It does not stop it. The mainstream explains the Atacama findings as consistent with an ancient, geographically restricted transmission cycle involving wild animal reservoir hosts and triatomine species now locally extinct. The disease was always present, they say. Poverty and substandard housing simply amplified transmission in the modern era. A reasonable position as far as it goes. But if Trypanosoma cruzi was circulating at detectable levels in the driest desert on Earth 9,000 years ago through transmission pathways that no existing model can account for, then the geography of this disease is a guess. A modern snapshot mistaken for a complete picture. Some researchers in ancient DNA and tropical medicine have begun asking whether the parasite's true prehistoric range was continental, whether it silently shaped the cardiac health of South American populations across thousands of years, and whether the demographic collapse that followed European contact was accelerated not only by smallpox, but by the silent failure of hearts that had been under attack for a generation or more before the first Spanish ship arrived. A disease that kills in 30 years is invisible to a civilization. It simply looks like people dying young. From a parasite that hid inside the heart for decades, we moved to a pathogen that hid inside history itself, erased from the textbook timeline even as its DNA was being pulled from the teeth of Viking-age skeletons in a Cambridge laboratory. The virus that predates civilization. Smallpox killed approximately 300 million people in the 20th century alone. It is the deadliest disease in recorded human history by almost any measure. We eradicated it in 1980 and locked the last 2 known samples in a pair of freezers, one at the CDC in Atlanta, one at the Vector Institute in Novosibirsk. We declared the problem solved. What we never seriously asked, until a team of researchers did so in 2020, is what smallpox was doing for the 5,000 years before it entered the historical record. The answer turned out to be more disturbing than the disease itself. In 2020, evolutionary biologist Barbara Muehleman and her colleagues at the University of Cambridge published a landmark paper in the journal Science. They had extracted ancient DNA from the dental pulp of 13 individuals spanning a 3,500-year window. The specimens came from archaeological sites across Russia, Siberia, Scandinavia, and the United Kingdom. Viking Age burials, Bronze Age graves, sites from the late Neolithic, and in several of those ancient teeth, the team found recognizable variola virus. Smallpox. The oldest confirmed specimen dated to approximately 3,700 years before the present. Not 3,000 years before the present, which was the previously accepted textbook date based on the pustular rash identified on the mummy of the pharaoh Ramesses V. 3,700. 2,000 years earlier than the history books said the disease existed. That alone would be remarkable enough. But the structural finding was the one that changed everything. When Muehleman's team reconstructed the ancient genomes and compared them to medieval and modern variola strains, they found a critical divergence in the gene responsible for suppressing the host immune response, a protein called CRMB. In the ancient strains, this immune suppression machinery was significantly less developed than in the version of smallpox that circulated through the early modern world. The ancient pathogen was, in all probability, less lethal, milder, more like a cousin to the monster that would eventually come. Here is where the timeline becomes genuinely unsettling. The mutation conferring maximum virulence to smallpox is estimated to have occurred somewhere between 1580 and 1650 CE. Not in antiquity, not in the ancient world. In the precise window of the European age of exploration, the period when ships were crossing oceans and making first contact with populations that had never encountered the pathogen in any form. The smallpox that arrived in the Americas and killed, by some estimates, 90% of indigenous populations in certain regions was not the same organism that had been circulating in Eurasia for 3,000 years. It was something that had just undergone a critical biological transformation. It had, in evolutionary terms, upgraded itself, and then immediately met the most immunologically naive population on Earth. The mainstream reads this as standard host pathogen coevolution. Pathogens evolve toward greater virulence when they encounter new, naive hosts. The timing of the virulence mutation is notable, but may reflect sampling gaps in the archaeological record rather than a sudden transformation. The ancient strains were attenuated, they explain, because they had co-evolved with their Eurasian hosts over millennia. The American catastrophe was a meeting of extremes: maximum pathogen virulence, zero host immunity, and the timing was coincidence. But if smallpox was circulating across Eurasia for 3,000 years before its virulence mutation, then every assumption we have made about how the disease shaped ancient history is built on the wrong organism. The smallpox that may have killed Ramesses V was not the same disease that killed the Aztec emperor Cuitlahuac. We have been treating them as the same thing, projecting a single pathogen profile backward and forward across 5 millennia. The Muehleman study suggests they were different organisms at different points in the same evolutionary trajectory. And then there is the question with no clean answer. Variola has no identified animal reservoir. Unlike almost every other major human pathogen, it appears to have had no wildlife host to retreat to between human outbreaks, which raises the question of how it survived across millennia and whether the reservoir we have not found still exists somewhere unsampled in the biological world. The two freezers in Atlanta and Novosibirsk may not be the last smallpox on Earth. They may simply be the last smallpox we know about. From the frozen north, where Viking teeth hid a killer for millennia, we travel to the fever dreams of ancient India, where someone, impossibly, wrote down the clinical symptoms of malaria in a sacred text 3,000 years before the parasite was identified under a microscope. The sacred fever. In approximately 1500 BCE, a Sanskrit priest composed a hymn to a fever entity called Takman. He described recurring heat and chills alternating with devastating precision. He noted the timing of the fever spikes and the association between the illness and the arrival of monsoon season and standing water. He prescribed treatments using specific plants. He documented the disease with a clinical specificity that should not have been possible. He just thought it was a demon. The Atharva Veda is one of the four foundational texts of Vedic tradition, composed roughly between 1500 and 1200 BCE on the Indo-Gangetic plain. Among its hymns is an extended address to Takman, a fever being of terrifying power. Vedic scholar Kenneth Zisk, in his 1985 study Medicine in the Vedas, was among the first to note that the Takman description aligns closely with Plasmodium falciparum malaria. Not just with fever in general. With the tertian pattern specifically. The 72-hour fever cycle in which the temperature spikes, breaks, allows a period of apparent recovery, and then spikes again. This is the diagnostic signature of falciparum malaria, the most lethal of the four main Plasmodium species. The Vedic hymn describes it with enough precision that a modern clinician reading it cold could identify the disease. Ronald Ross confirmed that malaria was transmitted by Anopheles mosquitoes in 1897, a discovery that won him the Nobel Prize. Charles Lavrin identified the Plasmodium parasite in human blood in 1880. The Vedic hymns predate both discoveries by more than 3,000 years, and yet the ancient Indian physician Charaka, writing approximately 300 BCE in the Charaka Samhita, explicitly attributes Takman fever to bites from tiny flying creatures born in marshes during the rainy season. A description of Anopheles mosquitoes, written 2,200 years before a Scottish physician with a microscope confirmed the same mechanism and was celebrated for the discovery. The pharmacological record deepens the anomaly further. The plant Saptaparni, known scientifically as Alstonia scolaris, appears in Vedic prescriptions for Takman. Modern pharmacology has confirmed its anti-malarial alkaloids. The Sushruta Samhita, composed around 600 BCE, describes malarial fevers in 5 distinct clinical subtypes that correspond, with remarkable accuracy, to the modern classification of 4 separate Plasmodium species, organisms that Western science did not distinguish as separate until the late 19th century. The ancient physicians were not guessing. They were observing something, categorizing it, and treating it with compounds that actually worked against it without ever knowing the mechanism. A 2001 molecular clock study by Joy and colleagues published in Science used genetic analysis to date the origin of Plasmodium falciparum to approximately 10,000 BCE in sub-Saharan Africa, with its spread into South Asia coinciding with the expansion of rice cultivation and the forest clearance that created standing water habitats across the Ganges plain. The parasite and the civilization grew together. The disease was endemic across the region for thousands of years before anyone wrote it down, and when they finally did write it down, they got it more right than they had any rational basis for doing. The mainstream explanation is generous and not unreasonable. Keen observation over generations can reveal empirical truths without requiring a theoretical framework to explain them. Farmers notice that fever follows the rainy season. Physicians notice that certain plants reduce fever. The tertian pattern becomes recognizable through sheer repetition. None of this requires the germ theory of disease. It requires only careful people paying close attention across long periods of time. But pattern recognition in a pre-germ theory world should produce ritual and supplication, not functional pharmacology. The Vedic texts do not just describe Takman. They prescribe treatments using plants whose anti-malarial chemistry we validated in the second half of the 20th century. That is not a consequence of watching people get sick during monsoon season. That is a medical tradition sophisticated enough to identify active compounds by their effects over thousands of iterations. And the question that no mainstream historian has answered satisfactorily is where that tradition began. Because if the Vedic texts are themselves already a codification of something older, already a writing down of knowledge that predated the Aryan migrations into the subcontinent, then the Atharva Veda is not the origin of Indian medicine. It may be an echo of something that once existed in the Indus Valley cities, in the urban civilization that built covered drainage systems and public water infrastructure 2,000 years before Rome, and whose writing we still cannot read. From a disease described in sacred text, we move to one carved in the human body itself, a bacterium that found its host 4,000 years ago and then stopped evolving because it had already achieved something close to perfection. The bacterium that stopped evolving. Most pathogens operate on a simple imperative: replicate, mutate, adapt. The evolutionary arms race between microbe and immune system is constant, relentless, and measured in generations so brief that antibiotic resistance can emerge in a hospital ward over the course of weeks. Mycobacterium leprae looked at that strategy and decided it was unnecessary. It found the human body somewhere around 4,000 years ago. It made itself at home, and then it essentially stopped changing. It is still here, it is still the same. And modern medicine still has no idea how it got inside us in the first place. In 2009, paleopathologist Gwen Robbins-Shug and colleagues published a study in PLOS One documenting skeletal evidence of leprosy in a human individual excavated from Balathal, a site in Rajasthan, India, associated with the late Indus Valley civilization. The skeleton showed periosteal reactions, resorption of the anterior nasal spine, and bone changes in the feet consistent with advanced lepromatous leprosy, the most severe form of the disease. Radiocarbon dating placed the individual at approximately 2000 BCE. It was, at the time of publication, the oldest confirmed skeletal evidence of leprosy ever found, pushing the disease's confirmed origin back by approximately 1,500 years. That finding was significant enough on its own. Then the genomics caught up. Stuart Cole's team at the École Polytechnique Fédérale de Lausanne published the complete Mycobacterium leprae genome in Nature Genetics in 2001, and what they found was genuinely strange. Roughly 50% of the genome consists of pseudogenes, non-functional remnants of genes that once worked but have been switched off and preserved as evolutionary debris. This degree of reductive genomic evolution is nearly unique among bacterial pathogens. It suggests that the organism completed its adaptation to the human host so thoroughly at some point in the distant past that further evolutionary investment became pointless. It had already solved the problem of living inside us. There was nowhere else to go. The comparative genomics that followed confirmed what that genome implied. A study by Monod and colleagues published in Science in 2005 conducted a global phylogeographic analysis of Mycobacterium leprae strains from patients across India, Africa, Europe, and the Americas. The mutation rate of the leprosy bacterium is among the lowest of any known organism. Ancient strains extracted from medieval European skeletons by Verena Schunemann's team, published in Science in 2013, were genomically near identical to strains circulating in patients in the same geographic regions today. The leprosy that infected a medieval peasant in England in the 12th century CE was, in any meaningful biological sense, the same organism that infected the Balathol individual in 2000 BCE. Over 4,000 years of continuous human infection in radically changing environments with radically changing host immune landscapes, the bacterium barely moved. The World Health Organization reported approximately 200,000 new leprosy cases globally in recent years, primarily concentrated in India, Brazil, and Indonesia. Exactly the regions where the oldest skeletal evidence appears. The disease has never left the geographic zones where it found us. It has simply waited. What makes the organism genuinely resistant to study is a biological stubbornness without parallel in modern microbiology. Mycobacterium leprae cannot be cultured in standard laboratory media. It can only be grown in armadillo tissue or mouse foot pads. It refuses to be studied on anyone else's terms. Even the questions researchers want to ask about it, about its origin, its transmission history, its evolutionary relationship to the human immune system, are constrained by the organism's refusal to behave the way a bacterium is supposed to behave. The mainstream explanation for the near-zero mutation rate is mechanistically sound. Slow replication, roughly once every 12 to 14 days compared to 20 minutes for Escherichia coli, combined with a massive pseudogene load, means there is simply less genomic machinery available to generate variation. The organism replicates slowly in a stable host environment, so evolutionary pressure is minimal. This is biology, not mystery. But a bacterium so perfectly adapted to human biology that it stopped evolving 4,000 years ago is, functionally, a parasite that found a permanent home. It did not conquer us in an epidemic sense. It settled in. It accommodated itself to our immune responses so completely that our bodies learned to coexist with it rather than eliminate it. A détente struck in the Bronze Age that has held uninterrupted to the present day. Medicine fights bacteria that evolve resistance in weeks. Leprosy does not need to. It already won that negotiation in a time before written history, and we have never renegotiated. From a bacterium that refuses to change, we cross to a disease that refused to stay, one that appeared from nowhere, killed thousands in hours, and then vanished so completely it left no biological trace in any laboratory in the world. The killer that disappeared. In the summer of 1485, a disease arrived in England with Henry Tudor's army and began killing healthy adults within hours. Not days. Hours. Within weeks it had reached London. Within months it had killed thousands. And then, as abruptly as it had appeared, it vanished. It came back 4 more times over the next 66 years, and then, in 1551, it was gone. Permanently. No descendant pathogen. No confirmed cause. No biological sample from any victim survives anywhere in the world. Medicine still does not know what the sweating sickness was, and in some important sense, it may never know. The disease, called Sutor anglicus in Latin and the sweating sickness in English chronicles, was first documented arriving at Milford Haven, Wales, on the 7th of August, 1485, among the mercenary forces accompanying Henry Tudor on his campaign against Richard III. It struck London within weeks of the Battle of Bosworth Field. English physician John Caius published the first systematic clinical description in 1552 in a book titled A Boke or Counsel Against the Disease Commonly Called the Sweat. What he documented was unlike anything in the modern medical catalog. The sequence of onset was consistent and terrifying. Sudden terror, severe headache, pain in the neck and shoulders. Then sweating so profuse and so sudden that contemporaries described it as though the body were melting from the inside. Then either recovery, almost always within 24 hours, or death. The window was that narrow. 2 Lord Mayors of London died within a week of each other in the 1485 outbreak. Anne Boleyn contracted the disease in 1528 and survived. Cardinal Wolsey contracted it in the same outbreak and recovered, though George Cavendish noted that at one point in the royal court that summer, more than 40 people were simultaneously afflicted. The case fatality rate in peak outbreaks is estimated at somewhere between 30 and 50%, based on analysis of parish death records from affected English towns. 5 distinct epidemic waves are documented. The 1485 and 1508 and 1517 outbreaks were confined to England. The 1528 epidemic broke that pattern catastrophically, jumping from England to Hamburg, then moving through Lübeck, Marburg, Strasbourg, and Basel, documented in records as far east as Krakow. The transmission speed implied by its movement across the continent, in an era of foot travel and slow river shipping, is extraordinary for any respiratory pathogen. Then the 1551 outbreak struck England one final time, and the disease disappeared from the historical record forever. Modern medicine has proposed candidates. Thwaites and colleagues argued in Lancet Infectious Diseases in 1997 for hantavirus, noting the rapid cardiopulmonary collapse in previously healthy young adults that characterizes both the sweating sickness and modern hantavirus pulmonary syndrome. Paul Heyman developed this hypothesis further in 2001. Others have suggested a variant of relapsing fever caused by Borrelia bacteria, an early influenza strain, or anthrax. Every candidate has been contested. None produces an identical symptom profile. None explains the geographic restriction to England in 4 of 5 outbreaks and the explosive continental spread in the 5th. None explains the 24-hour mortality window, and none explains the silence after 1551. The mainstream position is pragmatic. The sweating sickness was likely a variant of a known organism associated with a specific animal reservoir, probably carried by Breton or Norman soldiers in Henry's army. And when the transmission chain from that reservoir was eventually broken, the disease lost its ability to reach human populations. Animal reservoirs collapse. Transmission chains end. It is a documented mechanism. It is not impossible. But if it was a hantavirus, hantaviruses do not disappear. They persist in rodent populations for centuries. If it was influenza, it should appear in retrospective serological data from surviving bloodlines. Neither is the case. No biological material from any identified victim has ever been located in any museum collection or archaeological site. The sweating sickness left nothing behind except testimony. The accounts of physicians and courtiers and clergymen watching people die in hours, writing it down in a panic, and then watching it vanish as though it had never been. A minority of researchers, including John Wiley and L.H. Collier, in a 1981 analysis, have proposed that the pathogen may simply have driven itself to extinction, killing faster than it could transmit to new hosts, a biological dead end that burned out rather than persisted. If they are right, the sweating sickness is not a mystery waiting to be solved by identifying a surviving organism. It is already gone. It existed. It killed. And it left nothing that science can touch. From a disease that left no trace in biology, we moved to one that left a trace in everything. A fungal chemistry that for thousands of years drove entire communities to convulsions, visions, and the conviction that they were being attacked by invisible forces because they were. The devil's grain. For most of human history, entire villages would suddenly erupt in seizures. People would report burning sensations moving through their limbs, hallucinations vivid enough to shatter the boundary between the seen and unseen worlds, and then the blackening, the dry gangrene that would take fingers and toes and hands, leaving the skin dark and desiccated as charred wood while the person remained alive inside it. Priests called it divine punishment. Physicians called it bewitchment. It turned communities against their neighbors, filled convents with apparently possessed women, and provided the raw material for some of the most consequential acts of institutional violence in European history. It was the bread. The organism responsible is Claviceps purpurea, a fungus that parasitizes rye and other cereal grains by replacing the seed kernel with a hardened purple-black mass called a sclerotium. Inside that mass are alkaloids of extraordinary pharmacological potency: ergotamine, ergonovine, and lysergic acid, the direct chemical precursor to LSD. Eat contaminated grain over a period of weeks and those alkaloids accumulate in tissue. They constrict blood vessels, causing peripheral circulation to collapse and extremities to die while the heart continues beating. They trigger violent uterine contractions. They induce hallucinations complex enough to produce complete alternative realities. They create a syndrome with two distinct expressions: convulsive ergotism with its seizures and visions, and gangrenous ergotism with its burning and blackening, depending on the dosage, duration, and the victim's individual neurochemistry. Mary Kilbourne Matassian, in her 1989 book Poisons of the Past, documented ergot sclerosia in Neolithic grain storage vessels from Thuan in Switzerland, dated to approximately 3,500 BCE. The moment humans began cultivating rye at scale in Central Europe, they were unknowingly growing a chemical weapon in their fields. They had no means of seeing it. The sclerotia look like dark rye kernels. Ground into flour, they are invisible. The first recorded epidemic in a Frankish chronicle from 857 CE described a great plague of swollen blisters consuming the people. It would recur across the Rhine Valley and beyond for the next 1,000 years, killing tens of thousands. In 994 CE, chronicles attributed to Flodoard of Reims describe a single outbreak in Aquitaine, killing estimates in the tens of thousands. The condition became known as St. Anthony's fire, named not because of any theological significance, but because pilgrims traveling to the shrine of St. Anthony in Vienne, France, experienced relief. The relief came from leaving their contaminated local grain supply and eating clean grain along the pilgrimage route. The mechanism was purely dietary. No one understood this at the time. In 1976, scientist Linda Caporael published a paper in Science arguing that the 1692 Salem witch trial hysteria was triggered by convulsive ergotism in the Salem village rye crop. The behavioral symptoms of the afflicted girls, convulsions, burning sensations, the sensation of being pinched by invisible hands, visual and tactile hallucinations, matched the clinical presentation of ergotamine poisoning with precision. The rye cultivation conditions in Salem Village in 1691 and 92 were ideal for fungal contamination. Cold, wet winters followed by warm, humid summers, exactly the temperature profile that Matassian demonstrated correlates with documented ergotism epidemic clusters across Europe, produce maximum fungal growth. Historians of Salem have contested Caporael's hypothesis on social and documentary grounds. The medical hypothesis has never been refuted. Here is the modern irony. Albert Hofmann synthesized lysergic acid diethylamide, LSD, in 1938 at Sandoz Laboratories by modifying ergotamine. His first accidental self-intoxication in 1943 produced the same hallucinations, the same dissolution of perceptual boundaries, the same terror and wonder that medieval village chroniclers described in Survivors of the Fire. The same compound. The medieval villagers did not receive a controlled dose in a laboratory. They received it mixed into daily bread across weeks and months in quantities that accumulated silently until the body could no longer contain the effects. The pharmacologist E.Y.A.R. Gordon Wasson, working with Albert Hofmann himself, and classicist Karl Ruck, proposed in 1978 that ergot-contaminated grain may have been the active ingredient in the kykeon, the ceremonial drink consumed at the Eleusinian Mysteries, the most important religious ritual in ancient Greece, attended annually for 2,000 years by figures including Plato, Cicero, and Marcus Aurelius. If the Eleusinian initiation was pharmacologically induced by ergot alkaloids, then the philosophical architecture of Western civilization, Platonic idealism, Socratic transcendence, the immortality of the soul, may have been constructed on the experiential foundation of a fungal poison that was simultaneously, in less controlled contexts, burning peasants' hands off across northern Europe. We do not ask that question in the curriculum. It does not fit inside the story of steady upward progress. So the fungus gets a footnote, a box labeled agricultural history, and the philosophical implications are quietly set aside. From a chemical in the grain supply that drove civilizations into madness, we moved to something that did not need a grain supply at all. A disease written directly into the genetics of an entire ethnic group, the biological scar of an epidemic that ended centuries ago and is still with us today. The genetic scar. There is a mutation in your DNA, if you have Celtic ancestry, that will slowly fill your organs with iron until they fail. Your liver becomes cirrhotic. Your heart develops arrhythmia. Your pancreas calcifies into a diabetic organ. Your joints swell and lock. The process takes decades, developing quietly in midlife while you attribute each symptom to aging, to stress, to something else. By the time it is identified, damage is often already done. It is the most common, potentially lethal single-gene disorder among people of Northern European descent. Natural selection should have eliminated it from the gene pool 10,000 years ago. Instead, it spread. Something in your ancestors' past was so catastrophic that carrying a mutation for slow organ failure was still a better evolutionary bet than facing it without protection. The condition is hereditary hemochromatosis, caused by mutations in a gene designated HFE, most commonly a variant known as C282Y. Individuals who inherit two copies accumulate iron in the liver, heart, pancreas, and joints at a rate their bodies cannot regulate. Jeff Fetter and colleagues mapped the gene and published their findings in Nature Genetics in 1996. The carrier frequency in Northern European populations is approximately 1 in 10. Among people of Irish descent, the rate climbs as high as 1 in 7. Roughly 1 in 200 inherits two copies and is at meaningful risk of organ failure. By any standard of population genetics, this is an extraordinary number for a mutation with potentially fatal consequences. You do not get to 1 in 10 carrier frequency by accident. The evolutionary logic is straightforward in principle. For a harmful recessive mutation to reach that frequency in a specific population, it must have provided a survival advantage large enough to outweigh its costs. And the advantage must have operated specifically in that geographic population, since the mutation is essentially absent in African, Asian, and Middle Eastern populations. Something was killing Northern Europeans at catastrophic rates. Carriers of C282Y survived it better than non-carriers, and so the mutation spread. The leading hypothesis for what that pressure was published by Destante and colleagues in Blood Cells, Molecules, and Diseases in 2004. The argument is mechanistic and specific. Yersinia pestis, The bacterium that causes bubonic plague requires iron to replicate inside the macrophages of the immune system. It commandeers the host's iron storage proteins and uses them as a nutritional resource during the early critical phase of infection. The HFE mutation alters the iron-loading pattern of macrophages in a way that may impair this bacterial resource acquisition, making the cellular environment inside a carrier's immune cells marginally hostile to Yersinia pestis replication. In a disease with the Black Death's mortality rates, even a marginal reduction in bacterial replication efficiency could mean the difference between survival and death. And the Black Death killed between 30 and 60% of Europe's entire population between 1347 and 1353. The geographic specificity of C282Y is the detail that will not be explained away. Iron deficiency anemia. The other proposed selective pressure for the mutation, since it increases intestinal iron absorption, is a problem across all of Africa, South Asia, and the Middle East. If the benefit of the mutation was simply anti-anemia, it should have spread into exactly those populations. It did not. It spread in northwestern Europe in the populations that experienced the highest plague mortality in the 14th century and in subsequent waves throughout the 15th, 16th, and 17th centuries. The distribution of C282Y is not an anemia map. It is a plague map. Linkage disequilibrium analysis estimates the mutation first appeared in a single individual approximately 60 to 70 generations ago, roughly 2,000 years before the present, placing its origin in the Celtic Iron Age, long before the Black Death. This suggests the mutation may have been selected first by earlier Yersinia pestis epidemics, perhaps related to the Bronze Age plague lineages now being recovered from ancient DNA, and then massively amplified by the Black Death's extreme mortality. The people who survived the Black Death in Ireland and Scotland and Wales were not only the lucky ones, many of them, statistically, were the ones who happened to carry a gene that starved the bacterium trying to kill them. Their descendants are still paying the metabolic price for that survival advantage. Approximately 1 million Americans currently have hereditary hemochromatosis. Most of them have never been screened for it because medicine categorizes it as a relatively rare genetic disorder rather than as the living aftermath of the worst epidemic in European history. The Celtic curse, as it is informally known, is not a disease. It is a memorial. Encoded in the bodies of millions of people who have no idea they carry it. From a genetic scar of one pandemic, we arrive at the pandemic that may have predated all others, a form of plague pulling from Bronze Age teeth that bore no resemblance to anything in the medical textbooks. The plague before the plague. The Black Death killed 50 million people across Europe and Asia between 1347 and 1353. It is the deadliest epidemic in recorded history. For centuries we thought it arrived from nowhere. A catastrophe without prehistory, striking a medieval world that had no frame of reference for what was happening to it. Then, in 2018, a team led by evolutionary geneticist Eski Willerslev at the University of Cambridge sequenced ancient DNA from Bronze Age skeletal material and found something that changed the entire story. Bubonic plague wasn't the Black Death's origin point, it was the Black Death's descendant. Something older, and in certain critical ways, stranger, had been circulating through human populations for over 1,000 years before anyone wrote a single word about it. Willerslev's team, publishing in the journal Cell, extracted Yersinia pestis genomes from the dental pulp of individuals buried at sites spanning the Eurasian steppe, Siberia, and early European burial grounds. The specimens dated to approximately 3,000 to 2,500 BCE, roughly 1,500 years before the Justinianic plague of 541 CE, previously accepted as the first confirmed plague pandemic, and nearly 4,000 years before the Black Death. The ancient genetic material was recognizably Yersinia pestis. The same species, the same genus, unmistakable. But structurally, it was different in a way that rewrote the clinical picture entirely. The Black Death is transmitted primarily by flea bite. The flea acquires Yersinia pestis from infected rodents, the bacterium proliferates inside the flea's gut, and then the flea bites a human host and transmits the infection through its bite wound. This transmission pathway depends on a gene called YMT, Yersinia murine toxin, which allows the bacterium to survive the toxic environment of the flea's digestive system. Without YMT, Yersinia pestis cannot establish itself in a flea gut. Without flea transmission, there is no bubonic plague cycle. The Bronze Age strains identified by Willerslevi's team lacked the YMT gene entirely. They also lacked the PLA gene, plasminogen activator, which in the medieval and modern strains enables the bacterium to spread from the initial infection site through the bloodstream to the lymph nodes, producing the characteristic swollen buboes that gave the Black Death its visible signature. The Bronze Age pathogen was plague, but it did not travel by flea. It did not form buboes. It was something that we have no clinical category for, a Yersinia pestis that operated by different rules and spread through pathways that the modern disease model does not accommodate. The most likely transmission route for the Bronze Age strain, based on the genetic absence of flea vector machinery, was respiratory, person to person through droplets in the manner of pneumonic plague, the most lethal form of the modern disease. Pneumonic plague, untreated, kills in 90 to 100% of cases within 2 to 3 days. In a population with zero prior exposure and zero medical infrastructure, spreading through breath rather than flea bite, moving along the network of steppe migrations that were reshaping the genetics of an entire continent in the 3rd millennium BCE, what would that have looked like from the ground? The archaeogenomic evidence suggests it looked like the disappearance of a world. Studies of Neolithic European skeletal assemblages have documented catastrophic population declines in Western and Central Europe between approximately 3000 and 2500 BCE, with regional analyses suggesting population reductions of 50% or more within a few centuries. Conventional archaeology attributed this collapse to the arrival of Yamnaya steppe migrants, who replaced much of the genetic ancestry of Neolithic Europe within a remarkably short period. Wolfgang Hack, Johannes Krause, and their colleagues at the Max Planck Institute for Evolutionary Anthropology, working on the genomics of the migration, have proposed that this replacement was not purely demographic. Something cleared the landscape first. The Yamnaya did not just migrate into a continent. They moved into a continent that the Bronze Age plague lineage may have already partially emptied. A 2022 study in Science Advances by Raskovan and colleagues pushed the confirmed Yersinia pestis timeline even further, identifying the pathogen in a mass burial in Sweden, dating to approximately 5,000 years before the present. Before the Yamnaya migration reached that far north, suggesting the pathogen was already circulating in European populations through earlier trade and contact networks. The flea transmission gene, the mutation that would eventually give birth to the Black Death, is estimated to have evolved approximately 3,000 years ago in the window between the Bronze Age strains and the Iron Age. It was an upgrade, a single genetic acquisition that transformed a difficult respiratory pathogen into an organism capable of hiding in rodent populations indefinitely and exploding into human cities the moment conditions aligned. The mainstream does not contest these findings, The researchers who publish them are the mainstream. What they resist is the scale of the implication. A disease of undetermined clinical form spreading through unknown pathways, killing at unknown rates across populations with zero immunity, preceding the first recorded plague pandemic by 15 centuries and the Black Death by nearly 4 millennia. Not a contributing factor in a multi-causal collapse. A catastrophe distributed across centuries, moving just slowly enough that no single generation witnessed the full scope of what was happening to them. They simply saw their communities shrink. They buried their dead. They did not know the name of what was killing them because they did not have writing yet. And then the grass grew back over their settlements, and 1,000 years later, someone built a new civilization on the bones, and history officially began. 10 diseases, 10 entries from a medical archive that no standard textbook assembles in one place. And look at what connects them. Every single one breaks the story we tell ourselves about illness. That disease is a product of modernity, of the failures of civilization, of choices we made when we left the natural world behind. Prion disease was destroying Neanderthal brains 400,000 years ago. Cancer was attacking hominin bones 1.7 million years ago. Plague was circulating through Bronze Age populations 4,000 years before the Black Death gave it a name. These are not modern problems with ancient echoes. They are ancient constants that we have only recently developed the tools to see. The comfortable narrative runs in one direction, from ignorance to understanding, from darkness to the clinic, from demons to diagnosis. And that arc is real. The micro-CT scanner that revealed the Swartkrans osteosarcoma, the PCR that found Trypanosoma cruzi in Atacama mummies, the ancient DNA sequencing that pushed smallpox back to 3,700 BCE, these are genuine achievements of science. But the tools are only as useful as the questions they are aimed at. And the questions that get funded and published and taught are, by definition, the ones that fit inside an existing story. A 1.7 million-year-old cancer does not fit. A Bronze Age plague without buboes does not fit. A Celtic mutation that is simultaneously a lethal disorder and a plague survival mechanism does not fit neatly into anything. They get a paper, sometimes a press release, and then quietly sink into the literature, cited in footnotes, labeled as areas requiring further study. The sweating sickness gets no further study because there is no biological material to study. It simply vanished and left us the testimony of people watching it happen. The mainstream is not wrong to be careful. These findings are complex, and the implications are large, and large implications require proportionally large evidence. That is how science is supposed to work. But careful is different from incurious. And the pattern across these 10 entries is not randomness. It is a consistent undersizing of the medical past, an assumption that ancient populations experienced something simpler, something less, something that the trajectory of progress would eventually fix. The Vedic physicians who described the tertian fever cycle of falciparum malaria 3,000 years before Ross won his Nobel Prize were not doing something simpler. They were doing something different. The distinction matters. You are here because you noticed that distinction, because when the official answer landed and the subject moved on, your brain stayed behind and kept asking. That is not a small thing. That is the only mechanism that has ever actually revised what we think we know. This channel exists for that mechanism. So make it official, hit subscribe, and join the archive. Then go to the comments and tell us which of these 10 diseases disturbs you most. Not the most dramatic, the one that changes how you think about something. Make the case for your choice. The floor is yours. The bones do not lie. The genes do not forget. Every disease on this list left a mark, in fossilized lesions, in ancient DNA, in mutations still carried in living cells today. We look at prehistory and read silence, but the silence is dense, full of pathogens that burned through populations before anyone had a language to name them, full of epidemics that ended civilizations we never got to study, because the civilizations ended before the records began. The first entry on this list lives in your genome right now, a biological memory of something that destroyed brains long before the first city existed. The last entry moved through a continent and left it half-emptied, and the emptiness was called a migration. The question is not what these diseases were. The tools are finally sharp enough to answer that. The question is what else is in the record, waiting for someone to look carefully enough to see it. Subscribe.